Gracias. Buenas noches a todos. Tengo dos breves intervenciones en esta brevísima presentación. Y la primera es que quisiera recordarles que hace 75 años, en 1943, por decreto presidencial y por presión de figuras intelectuales muy importantes de los años 40, se creó el Colegio Nacional. Y desde entonces el Colegio Nacional tiene como sede aquí, este es el Colegio Nacional, en el centro histórico, e inicialmente el Colegio Nacional era el colegio del centro histórico porque aquí estaba el barrio universitario. Después el Colegio Nacional se volvió el Colegio de la Ciudad de México. Los profesores del Colegio Nacional fueron a impartir cátedra a las universidades y escuelas de la Ciudad de México y finalmente el Colegio Nacional se volvió realmente nacional. Es decir, que a lo largo y ancho de nuestro país, los miembros del Colegio Nacional imparten conferencias relacionadas con su quehacer intelectual científico. Y el mandato presidencial por decreto es difundir lo que nosotros sabemos. Como consecuencia del mundo en que vivimos, un mundo globalizado, el Colegio Nacional decidió que ya no es solamente nacional. Seguirá siendo el Colegio Nacional, pero tenemos que tener acuerdos con instituciones de otros países, y en particular con el Colegio de Francia. Y han ustedes de saber que el Colegio Nacional fue creado con una serie de principios que dieron pie al Colegio de Francia casi hace 500 años. Nosotros vamos por 75 años, yo no sé qué será de este mundo dentro de 425 años. Y esta es precisamente la razón por la cual el profesor Alain Prochiantz está con nosotros, hizo un largo viaje exclusivamente para inaugurar la cátedra del Colegio de Francia en el Colegio Nacional. Hemos establecido un convenio académico muy conveniente para las dos instituciones, de tal suerte que anualmente iremos 3 de nosotros a impartir conferencias en el Colegio de Francia, que tiene su sede en París, y vendrán 3 profesores del Colegio de Francia al Colegio Nacional y aprovecharemos para que ellos puedan conferenciar en las universidades de la Ciudad de México y eventualmente lo vamos a extender a la provincia porque es el Colegio Nacional. Entonces es un enorme placer tener con nosotros al profesor Alain Prochiantz. Diré unas breves palabras. Gracias a que él es actualmente el presidente del Colegio de Francia y que afortunadamente yo tuve el gusto de que nos conociéramos a principios de los años 80, porque coincidimos en el Colegio de Francia precisamente. Ahí trabajábamos, ahí nos hicimos amigos y es muy interesante que después de 38 años ahora estemos aquí en la Ciudad de México, en la sede del Colegio Nacional, también como yo lo hice para inaugurar la cátedra del Colegio Nacional en el Colegio de Francia. Él es un distinguido biólogo que se dedica al estudio de la morfogénesis y ha descubierto una serie de procesos, de moléculas que que hace algunos años era prácticamente impensable que los péptidos pudiesen viajar de célula en célula. Hacen cambios muy interesantes en el interior de las células, de tal suerte que tienen profundas implicaciones farmacológicas, sobre todo en la terapia celular o génica, si ustedes quieren. Y sobre todo él está muy interesado en cómo se forman los circuitos cerebrales y cómo eso que le costó tanto a la evolución crear, que es nuestro cerebro, encontrar las bases del funcionamiento de estas moléculas y de los circuitos cerebrales. Por supuesto, él es profesor del Colegio de Francia, es miembro de la Academia de Ciencias de Francia, de otras instituciones. Es ampliamente conocido en el mundo en el área de su trabajo y esta noche nos brindará su conferencia ¿Qué hay de joven en el viejo? Y yo creo que para todos nosotros el tema es sutilmente muy interesante. Alain, por favor, el micrófono es todo tuyo y por supuesto la sala del Colegio Nacional.
Well, you should have seen us 40 years ago. We were quite different. What's young and aging? First, I would like to say that I'm very, very pleased to be here. I mean, the second time in 18 months now. Last time was in January 2017, something like that, when we signed this agreement between Collège de France and Colegio Nacional. It's really, I mean, I've met so many incredible people here. I mean, and I hope we'll be able to exchange in the future, that many of you will visit us, and that many others will visit you, and that it will be just one community, I mean, as it should be between intellectuals across countries. It's true that I am a morphogenetician, Ranulfo, but when you asked me to give a talk on something of general interest, I thought that aging was, you know, the thing to do, because we are all interested in aging, okay? Well, aging is interested in us, I would say, rather. And I will not talk too much about morphogenesis today, but how morphogenesis can help us to understand aging in some ways. If I start now, you can see that what's young and aging has many meanings, and I'm not going to develop the meanings, but I hope that you will understand on the way. My first slide is taken from a review by Guido Kremer, as an Austrian working in France. And I will just concentrate on there are many aspects in aging. I mean, as you can see here, mitochondrial dysfunction, deregulated nutrient sensing, all the items that you can read on this slide. But I will concentrate today on genomic instability and epigenetic alterations. So I will try to make it, Ranulfo asked me to give a talk for the lay people, okay? I'm totally unable to do that. Though it's my job normally, right? But, you know, I still have a lot to learn on my own job. But if something is totally obscure, I mean, don't hesitate to raise your hand and say, well, Prochiantz, explain better, okay? So the first slide will show you a figure of how we see the chromatin today. I mean, the chromatin is a structure in the nucleus where the information is encoded. And there are two parts in chromatin. One is the closed chromatin, which we call the heterochromatin. And any gene which is encoded in this region is normally not expressed. It's silent, okay? And indeed, the silent genes in a fibroblast are not the same than the silent genes in a neuron. If not, there wouldn't be any difference between fibroblasts and neurons. And this is the open chromatin, which contains the genes which are expressed in a given cell. So all cells have a different structure of open chromatin and closed chromatin, euchromatin and heterochromatin. Now, in the cell, you need energy. And to make energy, you need mitochondria, okay? Now, each time you make a molecule of ATP, which is the energy molecule, you synthesize superoxides, reactive oxygen species, ROS, we call them. And indeed, when you breathe, when you are active, you make the ROS. And the ROS are extremely destructive. So you cannot live without destroying at the same time. Now, when you do this, when you synthesize this ROS, what happens is that you make breaks in your DNA, you make breaks in your membranes, you modify your proteins, and these bad guys have to be removed or repaired. And with time, unfortunately, it works less and less well. So that you have DNA damage, replicative stress, transcriptional noise due to the unfolding of the heterochromatin, and you have upregulated chromatin modifier, and this is your chromosome at the end, beginning and end, young and old, okay? At the bottom of this slide, I've showed the DNA damage story. So you make breaks in your DNA because of the reactive oxygen species, and these breaks have to be fixed. You have to repair your DNA. And you make breaks all the time, as you will see. Now, to repair DNA, you have two ways. One way is in dividing cells. This is a dividing cell, and in dividing cells, if you make a break on one strand of your DNA, you have the way to copy the other strand, which is intact, and thus to repair perfectly well. Now, if you do that in a cell that does not divide, and as you know, neurons do not divide, you cannot use this mode of fixing, this mode of repair, and you have to use this mode of repair, which is called non-homologous end joining. This is called homologous recombination. It is non-homologous end joining. So you make breaks, and then you have to fill the gap. And when you fill the gap, you make mistakes. You introduce mutations. And as you age, as you cut your DNA in pieces, as you repair your DNA, you make more and more errors. And at the end, you age, and at the very end, you die. Well, it's not really funny, where I start. Okay. And even with age, the repair systems are modified. You see here, this is the non-homologous end joining, the mode of repair that you have in neurons. And this is the old guys and the young guys. And as you can see here, the non-homologous end joining efficiency is decreased with time. So as you age, you repair less and less well. And this is why we age in part. And if you divide, you're not safe for that, because when you divide, each time you divide, you shorten a piece of DNA which is called the telomeres. And when your telomeres become too short because you've made too many divisions, the cells enter apoptosis. They start dying, and they disappear. So it's hopeless. I'm sorry to tell you that. We're not going to live forever. But breaking your DNA is something absolutely normal, okay? If you think, you break your DNA. If you do nothing, you stay alive. But why would you sleep your entire life? So each time you do anything, you will make breaks in your DNA. And this is an example of what happens in a mouse, okay? You take a mouse in a home cage. Now you put the mouse into a novel environment. So the mouse explores, the new environment. Have you seen a mouse in an open field, for example? It goes around, it looks, and after 5 minutes it goes into a corner and falls asleep. And as the animal is exploring the new environment, it makes breaks in its DNA. This is in the hippocampus, which is a structure in the brain that is responsible for the localization space analysis, to make it clear or simple. And as you can see here, in your parietal cortex, entorhinal cortex, in your hippocampus, here in your hippocampus, you look here how many breaks you make in your DNA. And this is a very healthy, it's a young, healthy mouse. But you take the mouse, you put it back into the home cage, and it fixes the breaks. You lose the breaks. So you repair. Everything is fine. You don't repair exactly the way you should. So even though you repair, there's something wrong in the repair. But you have plenty of time until, it goes totally wrong. Now, if you take a mouse in which you have introduced in the genome a gene mutated in Alzheimer's disease, for example, this is APP, it's amyloid precursor protein, which is a protein, a gene which is mutated in some forms of Alzheimer's disease, you see what goes wrong. You don't repair anymore. So it's not so much that you break your DNA, it's that you cannot repair it properly. And this is why these animals get sick at the end, or faster than a normal animal, okay? Another point I would like to insist on is that, people don't want us to touch the genome, genetically modified organisms. I mean, this is the evil. But, the genome modifies itself all the time. And it modifies itself because of the mobile elements that you have in the genome. What are mobile elements? Mobile elements are pieces of DNA which are transcribed into RNA, retro-transcribed into DNA, and insert into another place. Now, when you insert into another place, well, you introduce a mutation because you're not supposed to go there. 45% of the human genome is made of retrotransposons, of mobile elements. All these guys here are mobile elements, and they constitute 45% of the genome. Now, most of them are fossilized. They don't move anymore. But in the mouse, for example, you have 3,000 mobile elements that still move around in the genome. In the human, you have between 80 and 100 of these elements that still move in the genome and introduce new sequences, new breaks in many places. So the genome is not, even intact, is not a stable structure. It is unstable. In any living body, everything is unstable. As I was saying to the journalist a few minutes ago, we make new cells every year, and we replace our entire body. If you weigh, say, 800 kilograms, you make 800 kilograms of new cells every year. So you never face the same person, in fact, except for the nervous system, where you have some stability. Not total stability, but some stability. Now, these transposons, this is a line, these are the main family of transposons that are still mobile in the vertebrates. Now, here you have the transcription of this gene, the mobile elements, which goes to the cytoplasm, is re-imported, is translated, the two proteins ORF2 and ORF1 are translated. This complex of the proteins and the messenger RNA is transported back to the nucleus. You have a reverse transcriptase that transforms this RNA into DNA, and this DNA is inserted. It's inserted in a place, in a new place, and this makes a mutation in your genome. You can see it here. You have a break. The RNA comes here. It is transformed into DNA, and the DNA loops back, is introduced into the genome, and this blue guy here, this blue sequence here, is a sequence that corresponds to this moving mobile element. So it's important to understand that this activity, this mobile activity, this instability of the genome has something to do with aging. Take a fly. This is the brain of a fly. It's called the mushroom bodies because it looks like mushrooms. Well, I've seen better mushrooms than that, but it looks like mushrooms. So if you look at here, you have these mobile elements called gypsy. Why? Now, if you look at the aging fly, you see that the mobile elements are expressed more and more with time. They are expressed more and more with time because the heterochromatin is being destroyed by the oxidative stress, by the ROS. And you die with time. It's a fly normally, lives for 80 days, something like that. You see, this is a normal fly that dies with time. A certain percentage of the flies die with time. If you express or overexpress the mobile elements, as you can see here in a 30-day-old fly compared to a 14-day-old fly, you see that you accelerate the rate of dying of your animals. So if you increase this mobility of your genome, you will accelerate aging and death. Now, as I said, in the human, you still have 80 to 100 of these elements are still mobile. And they are all chromosomes. All the guys that stand up like that, red or yellow or orange, they are still moving. Okay, so on each of your chromosomes, you have sequences that encodes these mobile elements, which are transcribed, are going to reinsert in many places. And this is normal. This is life, okay? Life is not stable. We are not machines that sort of, decay with time. We are machines that reproduce ourselves all the time. This is what Claude Bernard called the silent embryogenesis. So we repair all the time, we destroy all the time, and these little guys, retrotransposons, are part of the destruction mechanism. They are also useful, as I will show you later. So this is a clear example, okay? Caloric excess, mitochondrial defect, ROS, oxidative stress, superoxides, and this introduces DNA damage. This introduces the induction of mobile elements, but also DNA damage per se, which is not always fixed. And unless you have good repair, when you go towards senescence and apoptosis, which is called aging. We have all experienced that now. I mean, most of you have had this experience, and even the youngest of you have had it. I'm sorry to bring the bad news. So you have this stress, superoxides, expression of lines, and this is useful because, it creates diversity in the genome. When it is in the germ cells, it creates diversity. So this diversity is subject to Darwinian selection, and then you select the gonads, the spermatozoids, primarily, which are most adapted, but at the same time, this adaptation, is selected by evolution. So this genetic variation, which is introduced by these elements, is selected through adaptation. This is a Darwinian processes. But at the same time, you have the bad aspect, which is DNA damage, senescence, apoptosis, and arrest, aging. This is the cost. So there is some good for the species, and there is some bad for the individual. This is the story of any living species. So I will tell you about a class of genes on which my lab has been working a lot for now a long period of time, which are called the homeogenes. What are homeogenes? This is a classical homeotic mutation. In this homeotic mutation, see, this is a fly head. In a fly head, here, you have an antenna. The antenna is like the nose of a fly. Now, in this mutant, the cells which have a program of transformation into an antenna believe that they are on the second thoracic segment of the fly. They are wrong, but they believe so because they don't read well the positional information in the genome. And for this reason, they develop like legs. And this is why this is called the antennapedia gene. So these genes are developmental genes, and they are the basis of our understanding of morphogenesis. How can we develop a program? Why, when you take a chicken egg, you will make a chicken. And if you take a human egg, you will make a human, even though it's one cell at the beginning. But in this cell, you have the entire developmental program. It's pretty simple, right? Now, these genes, they encode transcription factors, because the transcription factors have to read the program in the genome. They transcribe genes, which are the effectors of the morphogenetic program. So what we have discovered by accident, and it was pretty difficult to have people accept this idea, what we have discovered is that these transcription factors, a transcription factor is supposed to stay in the nucleus and to do the job in the nucleus to transcribe genes in the nucleus and nothing else, okay? So what we have found by accident, as I said, is that this family of transcription factors, which are called homeoproteins, because they are the products of homeogenes, because the homeogenes, when they are mutated, produce homeotic mutations, like the antennapedia mutation, that these transcription factors do something they should never do, okay? It's forbidden, but they do it. They transfer between cells. For example, this transcription factor here, the homeoprotein, is able to go out by a non-classical pathway of secretion. So it leaves the cell, it goes into the extracellular medium, and it is internalized by the next cell, by also a non-classical internalization process, on the basis of which we have discovered these peptides that Ranulfo was alluding to in his very nice introduction. Thank you, Ranulfo. Now, when this transcription factor goes inside the next cells, it regulates the translation of the messenger RNAs to make proteins. It goes to the nucleus, it regulates transcription, and it regulates also the epigenetic state of the nucleus, or the chromatin. Now, suppose that I take any cell in your body, anywhere, okay? And suppose that I'm able to tell which transcription factors of this family is expressed by this cell, and what quantity of this transcription factor is expressed by this cell, and I will be immediately able to tell you this cell is here, and not here, here. So that when I transfer these transcription factors from cell to cell, I'm also transferring positional information. So that the cell here that receives this transcription factor knows that this guy is in this position, because it combines with the other transcription factors here, so the combination tells the two cells whether they have to interact or not, okay? For example, I am in Paris, and I receive a transcription factor from Strasbourg or from Bordeaux, and I know that the guy is coming from Strasbourg and not from Bordeaux. It's very simple. I mean, it's obvious, okay? What was not obvious is that this transcription factor is in fact a morphogene, because it can travel between cells and transfer the information. Now, this is true during development. There are 300 proteins of this family. This is true during development, but it's also true in the adult. In fact, these transcription factors, they are expressed throughout life. Here, for example, this is a TX2, which is a gene. When you lose it very early, you lose the head, okay? This is engrailed. If you lose it very early, you lose a part of your brain that you call the midbrain. But in the adult, let's forget about this stage, in the adult, what you see here is that OTX2 is not expressed anymore in the cortex. It's expressed in other regions, the cerebellum, the ventral tegmental area, which is a place in the midbrain, in the dorsal midbrain, the superior colliculus. And engrail is the same. It's not expressed in the adult in the same positions where it's expressed in the developing animal. So these factors have signaling properties in the adult. They signal between cells. It's a novel mechanism for the passage of information between cells, how one cell talks with the other one. If you are interested in more, I would say, fundamental issues, I mean, you can come tomorrow to UNAM, where I will develop this, more thoroughly. Today, I'm going to tell you about a disease, which is called Parkinson's disease, okay? And the work I'm going to tell you about in as simple terms as possible, and again, if you need to interrupt me, you should not hesitate, please. This work was done by these colleagues, Yoko Arai, Olivia Baudouin, Julia Fuchs, who will soon become independent, Rajiv Joshi here, Kenneth Moya, Eugenie Pézé-Heitzig, Camille Ravel-Godroy, and these people have left the lab by now. This is Hossein Rikek, Colette Bouillot, François Gavier, Blodin de Thé, and Laure Saunier, okay, and has been published on the way. Now, what is Parkinson's disease? Parkinson's disease corresponds to the death of a class of neurons called dopaminergic neurons, which are in the ventral midbrain. They are here. And you can see here in the human, these cells are pigmented. This is a normal substantia nigra. It's called substantia nigra because they are black, they are pigmented. And if you are at a certain stage of Parkinson's disease, you see that the cells have disappeared. They are dead. And now you have this tremor, you have this difficulty to walk, all the syndromes of Parkinson's disease. Now, these cells of the substantial agra, SN, express this transcription factor, it's called NGREL, which is a homeoprotein transcription factor. And this is in the adult. You can see here by in situ hybridization, the messenger RNA encoding NGREL1 in the substantial agar, also in the VTA. And the NGREL2 messenger RNA, there are two NGREL genes, are expressed in the VTA. So, one thing is that you have to sort of realize is that even if you have a mutation that will, unfortunately, lead you to develop Parkinson's disease at the age of 50, 60, 70, you don't have the Parkinson's disease when you are young. Even if the gene is here at the beginning of your life, just after fecundation, okay? You have the mutation, but you don't have the disease. You get the disease when you have reached a given age. And this means that as you age, as you accumulate the oxidative stress, you become sensitive to the mutation that was present at the beginning. So aging is a risk factor for neurodegeneration. Not only because you get old, even if you have the disease at the beginning of your life, it needs a certain time of heterochromatin unfolding because of the oxidative stress to develop the symptoms. So what we did was to try to understand why we could express NGREL. We had to express NGREL in these cells. NGREL, as I said, is one of its transcription factors encoded by one of its developmental genes that I alluded to a bit earlier. So we created a mutant in which we remove one allele of NGREL1. So these cells, they have two alleles of NGREL2 and one allele of NGREL1 working. So you just miss 1 out of 4 alleles of NGREL, okay? And if you do that, as you can see here, with time, your cells in the midbrain, your dopaminergic neurons, die progressively as they do in Parkinson's disease, until you reach a level of loss where you start developing the symptoms. And the symptoms, it's a retrograde degeneration of your aminergic neurons in the target field, the striatum in particular, and you start having your motor behavior defects, and the non-motor behavior also, because it's not only a motor behavior which is affected. You also have an hedonism. For example, the mice don't like sweet water anymore. This is very grave. I mean, very, 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 very bad. If a mouse doesn't like sweet water, I mean, the mouse is very sick. People think they are depressed. Well, you know, who can tell that the mouse is depressed? It's pretty difficult, okay? But still, what is very interesting here is that if you miss only one allele out of 4, you have a slow death of your cells. Well, NGREL is a homeoprotein. So NGREL can transfer from cell to cell. And this we've shown in many studies before, but studies addressing developmental issues. Because it can be transferred between cells, it can be secreted, and can be internalized. So that if you inject NGREL in the midbrain, it will be internalized by the cells, okay? So what we did was to inject NGREL in the midbrain, and you see here in green that the protein has been internalized by the dopaminergic neurons in the midbrain. Now if you do that, you can save your cells. So this is what happens here. Here you inflict a very, very dramatic oxidative stress to the cells. You inject a drug which is called 6-hydroxydopamine, which is specifically taken up by the dopaminergic neurons and kills the cells immediately because of the huge oxidative stress that results from this capture of this drug, okay? The other cells stay intact because the capture of 6-hydroxydopamine is specific of this type of cells, the dopaminergic neurons. So you kill the cells, you see? You don't have them here, these nice cells that express tyrosine hydroxylase, which is the dopaminergic, the enzyme that is responsible for the synthesis of dopamine. You see, the cells are dead. But if you come just after that, and you inject NGREL, you save your cells. You can see here. You keep your cells. And this is forever. Forever, I mean, you know. If you believe in God, I will tell you that only God's love is forever. I mean, all the rest is just temporary, okay? I don't believe in God, I'm sorry, I mean, you know, I'm going to die. So, you see, you save your cells, and you save your cells forever. Why? Because you have an epigenetic effect. You restructure your chromatin. So you cells, which were old, the chromatin had unfolded because of the oxidative stress, and very rapidly unfolded because of the strong oxidative stress due to the capture of the 6-hydroxydopamine toxin. This chromatin comes back to a younger age. So this is pretty nice. It means that you can get younger, okay? This is reversing aging by adding a transcription factor that will restructure the chromatin from old to young. There are some good news sometimes in life, okay? So, let me get to the next point. When you do that, when you inflict the 6-hydroxydopamine stress, you modify the expression of the mobile elements. Because your mobile elements are repressed by the heterochromatin. I told you, I mean, in the heterochromatin, the genes are silent. So all the mobile elements which are in the heterochromatin are silent. Now, I destroy my heterochromatin. My lines are now free to express themselves. And as you can see here, this is the product of this gene here, ORF1P, which is one of the proteins encoded by the line element, and so you have an up-regulation of line expression, which is ORF1P, okay? Now, if you look at your Engel heterozygote, where your cells die, you have exactly the same thing. And this means that Engel protects against oxidative stress. Also, in normal life, okay? Now, if you lose your Engel during your life, it is as if you had a slowly accumulating oxidative stress. And this is why the cells die progressively in the mouse, which is missing one allele of Engel, one allele of Engel 1. You see, you have exactly the same thing in the mutant for Engel, and in normal cells, where you have the 4 Engel alleles, but in which you have injected the 6-hydroxydopamine drug. So Engel acts in 2 ways. It represses line expression through the maintenance of the heterochromatin structure, and it also directly represses line expression by binding to the promoters of the lines. The promoters in a gene is a region that regulates, controls the expression of the gene which is downstream of the promoter. Well, if this is so, blocking line activity or blocking line expression should save the cells exactly like Engel does it, okay? So this is the same experiment that I just showed you. 6-hydroxydopamine, Engel added, I saved my cells. Forever. Forever. I have exactly the same effect if I add stavudine to my animal, and stavudine is a reverse transcriptase inhibitor. It blocks the passage from RNA to DNA and blocks the insertion of the DNA in the genome. So you block the activity of your transposable element, your mobile element. The same if you modify the synthesis of ORF2P, which is one of the gene product of the lines, by introducing an siRNA which blocks the translation of this messenger RNA. And you can also overexpress PWE. PWE is a protein which blocks line expression. And as you can see, in the 3 cases, blocking line expression will modify the death of your cells, demonstrating that, as I showed you for gypsy in the fly, the mobile elements in the genome are responsible for aging, for cell aging and cell death. But the lines are normally expressed, okay? They are normally expressed, as you can see here. I mean, in fact, there is a basal expression of the line. So they are useful. They are useful. What is the use of these mobile elements? I mean, there are several things they do. One thing they do is to make nicks in long genes. You know, the long genes are extremely folded. So for the RNA polymerase to gain access to the promoters and transcribe the long gene, you have to make some nicks in the gene so that it relaxes. And then the enzymes which are responsible and necessary, like RNA polymerase 2 for transcription, can gain access to the promoters. It's also a function of another enzyme which is called topoisomerase. So the lines have a topoisomerase-like activity, and they facilitate the transcription of long genes. And long genes are extremely frequent in the nervous system. But the lines also have other functions. One of the functions they have is to serve as what we call long non-coding RNA. They hybridize with sequences in the genome, line sequences. They hybridize, the RNA hybridizes with these sequences, and they modify the splicing of the gene, they modify the methylation of the genes, they modify the expression of the gene. So this basic, I would say basal expression of this mobile element is physiological. Unfortunately, if you age, oxidative stress, you have the chromatin opening and you have too many lines expressed. And then you make too many nicks, and then you are beyond repair. And when you are beyond repair, it's not good news, because indeed, you're going to get sick and to die. To maintain line at the proper levels of expression, you have these transcription factors, NGREL, but also other transcription factors of the same family. But you can also have the normal line inhibitors. I just talked about stavudine, the reverse transcriptase inhibitor, and PIWI, this protein, which is necessary to maintain a low expression of the lines. Now I go back to this slide. Because there's another transcription factor, which is a TX2. I showed you a TX2 at the beginning. So TX2 is not expressed in the substantial alveoli. It is expressed in what we call the ventral tegmental area here. You can see it here. But OTX2 has the same effect as NGREL. It also, as you can see, saves the cells when they are jeopardized, when their survival is jeopardized by the injection of the toxins. So if you introduce NGREL in your cells, or if you introduce OTX2 in your cells, you save your cells. I come back to that. You can see here, this is what I showed you before. You have your line elements here, 17% of your genome. If you are expressed, this is what happens. You nick your DNA. Well, I guess I've done something wrong, because you're going to have the three things. Okay. Now let me go back to something a bit different. And this will be developed tomorrow at UNAM. This is called cerebral cortex plasticity. It's also an aging problem, okay? The work that I'm going to describe very briefly was done in collaboration with Takao Hensch, who is, I met Takao at the Weekend Institute in Tokyo, something like 10 years ago. And now he's moved to Harvard. And the people who've been working with me are Jessica, Namsuk, Ariel, Jérôme, Damien, and these have left the lab already, Clémence, Marine, Julien, and Clémentine. And this is Michel Volovich. Michel Volovich, I've been working with him my entire life. He was in the same lab where I did my PhD. I was working on RNA viruses, plant RNA viruses, you know. I went from the turnip yellow mosaic virus RNA to the brain. Big jump. And he was working on the plant DNA plant viruses. And we were good friends. We are still good friends. He's still working with me, even though he's emeritus. And a few papers on the way. No. Some of you may think that you are smart until you are 17, and then you become dumb. This is not the case. You are dumb for a period of time. You're smart for a small period of time, and you become dumb again, okay? So this very short period of time during which you are smart is called the critical period. I mean, for good reasons, because if you miss it, it's too bad for you. So there are many critical periods in the development of the cortex. For example, the sensory system develops earlier, so this is a conceptualization in the human. You remain open for a pretty long period of time. For numbers, you learn at 1, 2, 3 years. You are very plastic at 3 years, and then you are less plastic. And this is hearing, for example, in green. Hearing comes very early because you hear in your mother. When you're in your mother, you hear the noise outside. But you stop being able to learn sounds pretty early. That's why we cannot learn a foreign language very well when we are getting older. Vision, etc., etc. So the critical periods of the cortex are the period when your cortex can adapt to the environment. Okay? It's a very important period in life. And what we have discovered again was something very bizarre. I mean, I'm specialized in bizarre things for some reason, Could be some, I don't know why. What we have discovered is that this is for vision, binocular vision. Binocular vision, since Hubel Wiesel, is the paradigm used to learn cerebral cortex plasticity or to study cerebral cortex plasticity. Now, if you are a kid and you have a cataract, okay, the eye with the cataract doesn't see. So you cannot acquire binocular vision because only one eye is really seeing. The other one is very well connected to the cortex. You have everything. You have the ganglion cells that project to the thalamus to the layer 4 of the visual cortex. Everything is in place, but the eye doesn't see. So this eye doesn't learn how to see and develop as a lazy eye. But if you reopen the eye, if you remove the cataract, if you operate the kid before he's 6 or 7, the eye will learn again because your cortex will still be plastic. After 7, the cortex is not plastic anymore, so you cannot fix the disease. You will be amblyopic for the rest of your life. Well, it's not really important to be amblyopic, in fact. Binocular vision is not something absolutely necessary. There are 3% of the people on Earth who have amblyopia, okay? Now, what we have discovered is something which is amazing, is that when you open your eye, the mouse opens the eyes at P14, 14 days after birth. The eye is opened, and after 6 days at P20, OTX2, the transcription factor, increases in concentration in the cortex and accumulates in a specific class of cells, which are called the parvalbumin interneurons. The parvalbumin interneurons are inhibitory neurons. They inhibit the principal cells, called pyramidal cells, and this inhibition is extremely important because it allows the oscillations that take place in the cortex during cognition. But this is visual cognition, okay? Now, the maturation of these cells with time is associated with the accumulation of OTX2, and at P20, you open a period of plasticity. So if you reach the first threshold of protein in your cells, your cell becomes plastic. It can learn binocular vision. So the two eyes are open, OTX2 arrives in the parvalbumin neurons, and now you open a period of plasticity. It's a period where learning is possible, okay? Now, OTX2 goes on accumulating and reaches a second threshold, and then you close plasticity. So the same protein at two levels of concentration can open plasticity at P20 and close plasticity at P40, 40 days after birth. Okay, you're not totally lost? No. Maybe, but you're very polite. Now, what does that mean? It means that in the adult, the continuous accumulation of OTX2 in the PV cells maintain a non-plastic stage. OTX2 is not expressed by the parvalbumin neurons. It is imported by these cells, and it is imported from a structure which is in the brain. It's called the choroid plexus. You know, the choroid plexus is a thin epithelium which secretes the cerebrospinal fluid. I mean, it was called the kidney of the brain because it secretes the nutrients and it cleans the brain like a kidney does. Well, the kidney is very important. You know, you can live without brains, you cannot live without kidneys. Okay, I mean, there are many examples of that. So, during the entire life, you need this continuous import of OTX2 inside the PV cells to maintain a non-plastic stage. And indeed, it is very important not to be plastic. Because if you were too plastic, you would be stupid. You know? You would know who you are. You would know the person with whom you live. You would know where you parked your car. You know, you have no memory. So you need to have a non-plastic brain to maintain memory inside your cortex. So but the default state of the brain is plasticity. So you need this permanent arrival of the protein to maintain a non-plastic stage. So that if in the adult you remove OTX2, for example, by down-regulating the transcription of OTX2 in the choroid plexus, or by blocking the receptors which at the surface of the parvalbumin neurons trap the protein which is traveling from the choroid plexus, you get back to this zone here between threshold 1 and threshold 2, and you reopen plasticity. So you rejuvenate your cells. Again, I mean, there is a way to get younger after you have fixed the system through the permanent import of this transcription factor, because it is a transcription factor. And I will give you an example, okay? You can measure visual acuity. Now, this is a mouse, and this is a very bad experiment. I mean, a very mean experiment. It's not nice what I've done here, okay? We had a mouse, we had a very good visual acuity with the two eyes. And we closed the eye in the adult. One eye. At the same time, we reopen plasticity. And you lose visual acuity. So this means you have plastic because you can lose visual acuity in the adult. You know, when you go to bed, you don't wake up blind in the morning, normally. There is a nice experiment to do too, is to make an embryonic mouse by closing the eye during the critical period, like the cataract in the kid. So you close the eye during the critical period, and the mouse develops a lazy eye. And now, in the adult, you block OTX2 internalization, and the mouse recovers visual acuity. So this is one of very few examples of a cure for a neurodevelopmental disease, which is amblyopia. You can cure the mouse from amblyopia in the adult just by decreasing the amount of OTX2 that comes from the choroid plexus and is internalized by the parvalbumin neurons, okay? So you tell me, I mean, where is OTX2 coming from? Is it only coming from the choroid plexus? Well, I don't know, but there's something very weird. What is very weird is that this happens also in the retina. In the retina, OTX2 is expressed in the bipolar cells. It's a class of cell here. And goes into the ganglion cells that project in the thalamus that project into the visual cortex. If you block OTX2 in the retina, either using an antibody or siRNA, because you block the synthesis using the siRNA, you don't open plasticity. If you close the eye of your mouse at P29, at the peak of plasticity, you don't have the shift in ocular dominance. So this means that the transfer of OTX2 in the retina may have something to do with the opening of plasticity. So we injected OTX2 in the eye. Bam! And we found it in the parvalbumin neuron. So, in fact, the protein from the eye can go up, down, can go to the thalamus, and from the thalamus can go to the PV cells. When it goes to the PV cells, it forces the maturation of the PV cells and the synthesis of the receptors. The binding sites are glycosaminoglycans, it doesn't matter, because these transcription factors can travel along axons. And when you have the first receptors, now the protein that comes from the choroid plexus can be imported because the receptors are present. So this is why all critical periods do not open at the same time. You know, as I told you at the beginning, the critical period for language is not the same as the critical period for hearing or for seeing, okay? Because it depends on the first stimulus, which here is a sensory stimulus. So I open my eye, something happens in the retina that has consequences on my cortex, be it the transport of OTX2 or something else, could be the physiological activity of the cells. The receptors assembles at the level of the PV cells in the visual cortex. OTX2, which was floating around because it was released by the choroid plexus, is now internalized, and I start my gradient. I start my accumulation, threshold 1, threshold 2, and then I go down. At the same time, you know, I was asked to do it simple. Again, I mean, if you want to have the complicated story, come back tomorrow. At the same time, you have indeed an upregulation of the plasticity genes and a modification of the chromatin structure. Why do I say that? Why do I say that? Because, you know, amblyopia, you know, it's nice to have binocular vision. I cannot lie, especially when you use a microscope. But you can live without binocular vision very well, so, okay? But if you look in the cortex, and you look at OTX2 in the PV cells, in all the regions of the cortex, you see that OTX2 is in all PV cells throughout the cortex, which means that what we have shown for the visual cortex might be true for the entire cortex. And in fact, in a paper that we have recently published in molecular psychiatry, we showed that it's true for the auditory cortex. OTX2 arrival in the PV cells in the auditory cortex is responsible for the auditory cortex plasticity, and it's also true in the medial prefrontal cortex. In fact, you can teach a mouse to love music, complex music. If you take a mouse which is 60-day-old, a very old mouse, you know, these mice, they like silence. So you put them in an open field. On one side, there is a microphone with music. On the other side, there is silence. They go to the silent side, silent corner. But if you reopen plasticity in the adult and make them listen to Schubert, for example, for 2 weeks, they start learning how to like Schubert. And now they go to the place where Schubert is being played in the open field, in the corner with the music. So this means that this protein is responsible for plasticity not only in the visual cortex, but also in the auditory cortex and also in the medial prefrontal cortex. And this leads me to the 5 last minutes. I congratulate you for your patience. There is a critical period which is very important in the human, in fact in all animals. And this critical period is called, you remember? when the hormones arrive, it's called puberty. I mean, this is dramatic. It's a metamorphosis, okay? You don't look at people the same way. You don't look at yourself the same way. People don't look at you the same way. Something has changed, okay? Obviously, hopefully. This is a critical period. And you better adjust your brain, because if you don't adjust your brain to this change, well, you are in trouble. Now, most psychiatric diseases, schizophrenia, for example, starts between 15 years and 20 years of age. And this corresponds to the maturation of the PV cells in the dorsolateral prefrontal cortex, so that you have a shift of inhibition and excitation, which is the signature of the onset of plasticity, okay? Now, if something happens at this level, when you are flooded with the hormones, okay, then, as I said, you are in big trouble. So there is a hypothesis, which is that the etiology of many psychiatric diseases might lay in a bad discourse of the critical period in the dorsolateral prefrontal cortex. Because your GABAergic neurons, PV cells, do not mature, you don't have this interaction between the inhibitory and the excitatory neurons, and you are not able to create these oscillations, which the physiologists call the gamma bands. I will take you to a paper published by a guy called Wolf Singer, who is a great physiologist. And he's looked at the cognitive task, which is to recognize what we call a Monet picture. A Monet picture is a face like that, a Monet face, where it's so contrasted that it takes some time to realize that this is a human being, okay? Now, if you reverse the figure, it's even more difficult. So it takes time to give the correct response. So now, very early in the face condition, very early in life, it takes, say, 1,000 seconds for the child to say, this is a face. And then, as the human mature until early childhood, it takes less and less time. The child becomes smarter. He recognizes the face more readily and faster. But in late adolescence, it loses this ability, in part. And this can be seen here by the gamma bands, the oscillations. You see, the adults are very good. The early childhood is here, the late childhood is here, the early adolescence is in blue, and the late adolescence, bang, I lose my ability. Why do I lose my ability? Because I am remodeling my terminals in my dorsolateral prefrontal cortex. And then I have to go back to something normal in the adult. But if you're not able to go from gray to black, then you have a cognitive issue, okay? Is it a TX2? That's a good question. Thank you for asking. But if it is a TX2, where does it come from? Well, there is a structure in the brain that makes a lot of a TX2. And this structure is called the ventral tegmental area, okay? Be careful. This is a hypothesis that I'm developing here, okay? Now, the VTA here is a dopaminergic structure, like the substantia nigra, very close to the substantia nigra that I told you about when I was mentioning the Parkinson's disease. Now, this structure is full of a TX2. Now, the hypothesis that we make is that the TX2 is transported from the VTA to the prefrontal cortex, and possibly to the ventral hippocampus, to the nucleus accumbens, and to the amygdala, which are very much involved in mood regulation, and that this VTA plays a role that the eye might play in the visual system. I call it the mood eye, okay? And once OTX2 has arrived, it forces the maturation hypothesis. It forces the maturation of the receptors at the surface of the PV cells, which are now able to capture OTX2 from the choroid plexus. This would mean that amblyopia is a beautiful model for the study of mood diseases in the human, okay? And that all the tools you could develop to fix amblyopia might be useful to fix very complex diseases such as bipolar disorders, schizophrenia, and others. We don't have evidence for that, okay? But we have a few data. One data is that in the OTX2 heterozygote, I told you about the engrade heterozygote. In the OTX2 heterozygote, where you lack one allele of OTX2, your mice are hypo-anxious. They have a problem, okay? If you put a mouse on the elevated pulse maze like that, normally they are very careful. The mice don't like to go, no, they fear to fall. I mean, that's pretty normal, right? So they stay inside the box. If you have one allele of a TX2 removed, they go on the maze, they go on the platform. They are adventurous. So this has an effect on their level of anxiety. Okay. And as I told you, I mean, I just mentioned that we have issues with the auditory cortex and with the medial prefrontal cortex. Now, as I said, OTX2 is expressed in the VTA, which is, this is a substructure diagram where engrade is expressed. This is where OTX2 is expressed in the VTA. And a very nice paper came out of the lab of Eric Nestler a year ago. Eric Nestler is a colleague working at Einstein University in Columbia, okay, Albert Einstein. So you take a mouse, and the mouse has kids, okay, babies. Now if you remove the kids from the mother before P10, 10 days after birth, nothing happened. The kids will develop normally. If you remove the kids after weaning at P20, nothing happens. The kids will be normal adults. If you remove the kids between P10 and P20, maternal separation between P10 and P20, the kids will develop what we call a social defeat symptom. So that if we are confronted to an aggressive male, we will experience defeat. We will not love sweet water, we'll be depressed. And this period, during these 10 days, when you have separated the mothers from the offsprings, you have a transient decrease in OTX2 in the VTA, which comes back to normal after. But this small decrease in OTX2 concentration in the VTA between P10 and P20 is enough to mark the cells epigenetically for life so that the animals will be abnormal as adults, okay? And I come back with my mobile elements because, in fact, when you do that, you activate mobile elements. In fact, when you do that, you activate mobile elements in the hippocampus. This was just shown by Rusty Gage, a scientist working at San Diego at the Salk Institute. And there is a lot of elements in the literature showing that in people affected with neurodevelopmental disorders or schizophrenia, you have an abnormal expression of line elements. So that in fact, we may think that the two lines of research may sort of be reconciled. The decrease in OTX2 expression, the increase in line elements, so that you have some epigenetic marking of your VTA cells due to this expression, downregulation of OTX2, upregulation of mobile elements, that will make your animal sick for life. Is there a cure for that? I don't know, but it's very likely that these new transcription factors, this new mode of signaling, may open the way, we hope, to the understanding of very complex diseases and possibly open the way to novel strategies to take care of them. And I would end up by thanking the agencies that have supported my work and the work of my lab, Collège de France, indeed. Since I'm here, it has to be Collège de France. In some, CNRS and the European Research Council. Thank you very much. Are we supposed to take questions? Yeah? This is on you?
There are some simplicidad de cuestión en español.
Okay. You don't have to ask questions. There is a good buffet upstairs, I think. No? Yeah. You can speak in Spanish. Salvador, you will translate if I have a problem. The lady cannot hear you. Sí, venga. You want to come here and use my mic?
A ver, se menciona siempre, lo he visto muy repetitivo aquí, se menciona que en las investigaciones se toma un ratón o un chango, un mono, pero dicen uno, y normalmente en estadística la muestra de uno es nada, pero no sé si se pueda universalizar el uno en la investigación y decir, esto nos demuestra cuando estamos trabajando en una muestra de un ratón, y eso me llama mucho la atención, si es válido o es insuficiencia de que se tome un ratón, porque probablemente las conclusiones haya sesgos muy grandes.
Yeah, thank you for asking this question, but we don't take one mouse. Unfortunately, we take hundreds of mice. You know, what we show is statistics. So when I say the mouse, I'm talking about the species. Okay. Can you turn it on again, or? So you see all these little bars. Okay? All these little bars that you see here means statistics. It means it's the mean of experiments done here in this experiment for 12 mice. These are 4 mice, here are 7 mice. So in fact, you make statistics, and there are tools to tell you whether the statistics are right. So when you say here, P less than 0.001 means that you can be wrong by 1 out of 10,000, okay? It's a probability. So in biology, it's always a probability, but to be publishable in a result has to be, you have to give your statistics. So if I say P is 0.005, this means that you have 5 out of 1,000 chances to be wrong. You can be wrong. 5 out of 1,000, you can be wrong. But you have to indicate the statistic all the time. And this is always if not, you're not published, you know. Did I answer your question correctly?
Professor Prochiantz? Yep.
Yeah.
I have 2 questions, in fact. The first one is coming from the fact that homeotic genes, the homeotic locus...
If you speak in Spanish, no, the translation has to be in English, please.
Okay, I continue in English? Okay, que lo haga en español.
You speak English, okay, sure.
English, please.
Okay, the point is homeotic genes, homeotic locus possess a lot of transcription factors, a lot of genes all along the locus of the homeotic genes. Then my point is, why engrail is so specific to these line retrotransposition elements and not other transcription factors that could also affect other types of repetitive elements?
Well,
This
it's a
is
good point.
my first question.
It's a good first question. Can I answer the first question, or you want to give me
Sorry.
the second one?
Okay.
You want the second one?
No, go ahead, please.
Well, the first question is that we've been studying engrail 1 in these cells, okay? So I'm not saying that other transcription factors don't do the job. In fact, we know that OTX2 also does the job. It's possible that the promoters, which are recognized by engrail and repress line expression, are also recognized by other transcription factors. For example, sirtuins, which are known as anti-aging factors, can repress line expression. So in this case, we try to understand how the lack of one allele of engrail could be translated in DNA breaks.
Okay.
I didn't show you the DNA breaks. And we found that the lines were in the pathway. This does not mean that other transcription factors of this family of other families like sirtuins cannot do it.
Okay. And my second question is, part of the phenotypes that you are describing us are related to the degree of heterochromatization or loss of heterochromatin. When you reach some point of losing heterochromatinization, you should also have homologous recombination between all the families of repetitive sequences. And then which is a critical point to go to a high degree of chromosomal instability instead of going to a control, as you mentioned, apoptosis or senescence control?
Thank you. This is absolutely true. I mean, one of the effects of having the unfolding of heterochromatin that the lines can recognize themselves at long distances in the genome and then make crossing orders and remove a big piece of DNA between the two sites and add one into another site. So you have this huge genome instability, but this takes place only in dividing cells. Okay. Here we have postmitotic neurons, and you don't have homologous recombination in non-dividing cells. This is why you need non-homologous end-joining repair systems to repair your DNA.
¿Alguna otra pregunta, comentario? Pablo, aquí, adelante, por favor, el micrófono.
I was very much impressed by one of the last slides where you show maternal care and how important it is to have the breeding, the children together with the mother and all the after.
In the mouse. In the mouse.
Yes. My question was, of course, considering what's going on in the world with our neighbor, separating children from parents, to what extent we have scientific evidence, I believe we have, that this is really producing that kind of behavior and damage.
There is evidence for that people who have experienced bad experiences during their life, even without having children, have modified epigenetic states which sort of impinge on the health of the offsprings. We don't know how this works, because this would mean that the epigenetic modifications can be sort of made heritable across generations. But it is obvious that in our species, dramatic experiences like starving or the camps during the Second World War may have modified either the genome, possibly by modifying the expression of the mobile elements, and if this happens in the germline, it is transmitted to the descent, or epigenetically, and then how an epigenetic modification can be transported from generation to generation is something that takes place, but we don't know how. I mean, I've not seen any convincing experiment showing a mechanism for an heritable epigenetic modification, at least in vertebrates. I mean, in worms, in the Caenorhabditis, I mean, it can be held for several generations. There's a very nice experiment which was done by Susan Lindquist in the 80s. I mean, she took flies and she raised the flies with a drug called geldanamycin that inhibits a heat shock protein called HSP90. And most of the flies die, except a subpopulation of flies that survive. And they are weird, you know, they are monsters. There are hairs in the eyes, I mean, just monsters. And then they removed the drug. And the idea was that the phenotype would come back to normal. No. The monstrous phenotype was maintained. And nobody had a good explanation for that. People thought, well, this is the transmission of inherited traits, like a Lamarckian sort of a mechanism. Now, what had happened, and this is very interesting, that because of the stress, the inhibition of line expression was abolished in the germ cells, so that a lot of mutation occurred in the germ cells, the spermatozoids, in fact. And that by chance, one of these guys, or two or three, could have mutations allowing to survive at low activity of HSP90. But since this was in the germline, it was fixed genetically. It was a Darwinian selection operating at the level of the germ cells. And forever, these flies will have hairs in the eyes and a very, very bad aspect. You wouldn't like to have a fly like this with you. But it's a very good point. I mean, honestly, it's very likely that some things like that happen. Yeah, can happen. Yep.
Yeah, so I have two questions. The first one, it's related with the receptor that internalize these signaling transcription factors. How ubiquitously expressed is this? Can I find it in other cell types beyond neurons? I'm thinking if these signaling factors can travel to other cells. That's the first question. And the second one, you have shown that lines are problematic, right, in aging, and they have like these double-strand breaks. But Fred Gage have shown that they can also express in neural precursor cells, and it looks like in those early moments of neural development they are important. So where is the distinction between this damaging effect later in life and the possibility that they might be useful early in development for neural maturation?
So the first question was the receptors. Well, I wouldn't use the word receptors. So I did it to make things simple. So I would call them binding sites. Because you have a receptor, you need to have the transduction mechanism. So I would say binding sites. So Camillo Golgi, 100 years ago, described what he called the perineuronal nets, which is a very complex matrix which unwraps the parvalbumin neurons in the cortex. So what we discovered with Takao is that the binding sites for OTX2 is composed of complex chondroitin sulfate E and D, which are embedded in the PNNs. And these PNNs appear at the time of onset of the critical period. So this is for your first questions. You have PNNs in other places, but most PNNs are around the PV cells in the year 3-4 of the cerebral cortex. For the rusty stuff, Gage, Rusty Gage, Fred. Fred is working on dividing cells. So Fred has been working on progenitors in the hippocampus. So he's only been looking at the transposition in the hippocampus. Fred has never been working on postmitotic neurons. And in fact, until very recently, it was not accepted that mobile elements can be expressed in postmitotic neurons. In fact, they are. And their function is not only to be transcribed, retrotranscribed, and inserted. They can act also as long non-coding RNA, okay? And they don't have to be full length for that, okay? So when you transcribe from the 5' UTR your RNA, even if you don't go to the end of your transcription, this long non-coding RNA have functions that many long non-coding RNAs have. Say, the regulation of transcription, the regulation of splice, and also the regulation of methylation on the CPGs and the CPAs. In fact, in the brain, what you have is primarily CPAs and not CPGs.
Yo quisiera hacer un breve comentario antes de dar por terminada esta magnífica sesión. Hace unos meses, el País le hizo una entrevista al profesor Prochiantz y allí expresó el doctor Prochiantz qué era el Colegio de Francia. El Colegio de Francia dijo que no querían profesores en el Colegio de Francia que fueran a comprobar lo que otros habían hecho o estaban haciendo en la investigación, que querían tener individuos en la frontera del conocimiento. El trabajo del doctor Prochiantz a mí me parece fascinante porque, primero, lo que a la naturaleza le tardó, la evolución le tardó tanto tiempo en construir, y algo que nos parece muy sencillo, ellos están tratando de entenderlo a nivel de la maquinaria celular, internamente. Y esto nos lleva a una reflexión extremadamente interesante. ¿En qué momento, cuál es el momento apropiado para enseñar? ¿Cuál es el momento apropiado para retener? ¿Cuál es el momento apropiado para generar nuestras acciones que nos permiten interactuar? Pero él muestra que hay condiciones verdaderamente simples, aparentemente simples de la vida común y corriente, como sucede a la hora de parir unos ratoncitos. El solo hecho de separarlos en unos cuantos días de la madre puede generar conductas que se quedan para siempre en estos cerebros, que obviamente la conducta viene del cerebro. Entonces a mí me parece verdaderamente muy revelador todo esto, porque estamos por entender en qué están involucrados todos estos factores transcripcionales. Y una de las preguntas que me hice a la hora de estar escuchando al doctor Prochiantz, ¿qué tan lentos son estos procesos? Y son simplemente estos péptidos que se van moviendo a través de la sinapsis y van metiéndose a las células y después se van moviendo de circuito a circuito. ¿Qué relación guarda con la actividad de las neuronas? Que es lo que estudiamos muchas veces los neurofisiólogos, porque él mismo nos mostró un caso de las oscilaciones gamma, cómo están asociadas con ciertos momentos de maduración de los circuitos cerebrales, cuando muy probablemente es cuando hay un balance, excitación, inhibición apropiada para estabilizar el contenido de la información. Bueno, pues con esta charla damos por instalada la cátedra del Colegio de Francia en el Colegio Nacional y estaremos encantados de recibir a nuestros colegios del Colegio de Francia, de las diferentes disciplinas, del área de humanidades, astronomía, física, matemáticas, biología, medicina, y solo me resta más que agradecer la presencia de cada uno de ustedes y quisiera que despidiéramos al doctor Prochiantz con un aplauso. Gracias, buenas noches.